Good afternoon, everyone. Thank you so much for joining us today. My name is Brooke, and I'm delighted to welcome you to the Annovis webinar. Today, we will provide updates on the company's clinical progress and answer questions from the audience. Before we begin, I would like to remind everyone that today's call may include forward-looking statements. These statements are based on current expectations and assumptions and are subject to risks and uncertainties that could cause actual results to differ materially from those projected. For more information on the risks and uncertainties related to Annovis Bio's business, please refer to the forward-looking statements and the company's latest SEC filings. To guide us through today's presentation, I am pleased to introduce our speaker, Dr. Maria Maccecchini, the founder, president, and CEO of Annovis Bio. Today's presentation will address the progress of the pivotal Phase 3 Alzheimer's trial, the status of the Parkinson's program, and additional clinical directions. We also welcome our audience to leave a question in the comment section. There's a Q&A box on your toolbar. You can leave a question during the webcast or raise your hand by using the button on the taskbar. Once it's your turn, you will be unmuted and allowed to ask your question directly. Without further ado, I would like to turn the call over to Dr. Maria Maccecchini.
Good afternoon, and thank you so much for attending. As Brooke already said, I would like to give you first an overview of the company, where we are with all our clinical studies, how we use the clinical studies we have done to continue our development and go into pivotal studies in Alzheimer's and in Parkinson's disease. So first, the overview. Most of you may have seen this slide. It is our pipeline. And as you can see, in Alzheimer's, we are in the second study, large study, which we believe that when we will read out the data will be adequate to be the pivotal study and to actually allow us to file an NDA, one for symptomatic and one for disease modifying. In Parkinson's disease, we have finished one large study. And at this point, we are looking at opening an open label study for the patients that were in our previous study and for some additional patients that have deep brain stimulation. And finally, Lewy body dementia is a disease that is something between Alzheimer's and Parkinson's. Patients with Lewy body dementia exhibit the symptoms of an Alzheimer's person and of a Parkinson's person. So those three indications are the ones we are pursuing. For the rest, we will get there when we have more money. This is a very busy slide, but it actually shows how many studies we have done. We have treated way over 800 patients. And here, the first two studies show safety in healthy volunteers. The next two studies show very small biomarker studies and a little of efficacy in Alzheimer patients. The next two studies are two small studies, one in Alzheimer's, one in Parkinson's. Then we did a study, a large study, 350 study in Alzheimer's and a 400 or 500 patient study in Parkinson's. Lately, we have done all the studies necessary to file an NDA, which is a food effect, a combination or a bridge study between the old semicrystalline form and the new crystalline form of buntanetap, and an ADME study, which is you really give radioactive material to a human being and you look where the radioactivity shows up, where in the body, where in the feces, where in the urine. And then finally, we have the last study that we're conducting right now, which is the 760 early Alzheimer patient study. So as you can see, we have done a lot of studies and we have a lot of understanding of our drug. And I will try to walk you through to see what we know for sure and what we don't know for sure. So let's first look at cognition and safety. This is what we believe we are sure that it's correct. I told you we have two very small Alzheimer's studies. When we looked at cognition, and then mostly safety. So the first one on the left, the ADCS study, only treated 5 patients. But these 5 patients look substantially better than their placebo counterparts. Then our ADPD study only treated 9 Alzheimer patients. And again, these 9 Alzheimer patients looks a lot better than the placebo. When we combine the 2 studies, they were both 1-month study, we see that the 14 treated patients actually show highly statistical improvement in cognition over baseline and almost over placebo. So while you can discount these studies are too small, the fact that they both, and together even more so, show efficacy over placebo is very encouraging. Since we were encouraged, we did a 350 patient Alzheimer's study. And we took moderate and early Alzheimer patients, and we did not check for plaque and tangles in the brain. So what we found is that we had 40% patients that did not have Alzheimer's disease. We were able, after the fact, to figure out who had Alzheimer's disease by using the p-tau217 test. It's a test in plasma that tells you and correlates really well with plaques and tangles. So when we looked at early Alzheimer patients that had Alzheimer's and were early, we see the graph on the right. All 3 doses show statistical significance over placebo, and the 30 mg dose is over 3.5 points better than placebo. You will see that 3.5 points is a very nice improvement. But whenever you cherry pick, you want to have some safety net that in fact you're not cherry picking. And so we looked for robustness of our data by doing a forest plot. A forest plot looks at groups within your data and looks at how these groups behave. So the blue square on the top is the overall outcome. That is what I just showed you, that the 3.5 points improvement in cognition. But then we looked at age. Age makes no difference. Patients respond. We looked at APOE4 carriers. APOE4 carrier or not makes no difference. People respond. Heavy or light, people respond. Ethnicity, Hispanic, Latino or not, they respond to our drug. Gender, female or male, they respond to our drug. And then concomitant medication. On top of Aricept and Namenda, which are the current standard of care, people respond. Interestingly enough, if they are on antidepressants, they respond really well. And then if they take no concomitant medication, they also respond. And then finally, black and white. Every group we looked in early Alzheimer's response to buntanetap. That gives us some belief or some strong belief that the drug actually works in that population. Safety, it was important for us to show that buntanetap is safe, especially in APOE4 carriers, because the 2 novel approved drugs, Leqembi and Kisunla, have high side effect profiles in APOE4 carriers. So we looked at all concentrations and placebo. We looked at carriers and non-carriers, and there's absolutely no difference between placebo, carriers, and non-carriers. Our drug is safe no matter what genetic makeup the patients have. Well, let's look what else. In what other group we have seen cognitive improvement, and that is in Parkinson's. In Parkinson's, in the whole ITT population, what you see on the left is that the population, placebo, gets a little worse, and the 2 treated groups actually don't. However, when we specifically look at the Parkinson's patients that are cognitively impaired, we see a huge improvement from minus 1.7 to a little better than baseline. And it really shows that our drug improves cognition in Parkinson's patients. Again, we wanted to see the robustness of our data. And again, we did a forest plot. So here we looked what else do these mildly cognitive impaired Parkinson's patients respond to. And what we saw is that they respond to every single one of the Parkinson scales. UPDRS is a Parkinson scale, has 4 parts. They respond to part 1, part 2, part 3, part 4, CGIS. And then below, response is on the right, not on the left. And you see that they respond to WAIS coding. Of course, to MMSE, that's how we selected them. And then to PGIC. So again, we feel very confident that this is not cherry picking, that these patients that are cognitively impaired and have Parkinson's actually do respond to our drug. And again, safety. We wanted to see if the 2 doses and placebo have the same safety profile, and they do. So we have shown consistently that our drug works in cognition independent of Alzheimer's and Parkinson's, and it is safe independent in Alzheimer's and Parkinson's. Let's see how we use the data I just showed you to move into a pivotal phase 3 study. What we learned is that it works in early Alzheimer's. So we selected patients by MMSE over 20. High numbers of MMSE are better, low numbers are worse. MMSE 21 to 28 spans the gamut from early and mild. And if they have that MMSE, then we look to whether they are really Alzheimer patients. And we do the p-tau217 test in plasma. And then we do a volumetric MRI that actually is the baseline for us to then to continue the study. So let's see on how we look at efficacy for symptomatic and efficacy for disease modification. For symptomatic, we look at ADAS-Cog13 and ADCS-IADL. This is what the FDA told us to do. For disease modification, we choose biomarkers that may show disease modification, and then volumetric MRI. The biomarkers we have so far chosen, and we may increase the number as more biomarkers in plasma become available, are inflammatory ones like GFAP, are nerve health ones or nerve distress ones like neurofilament light, are p-tau217 and brain-derived tau. And as I said, as they become available, we will do more biomarkers. Volumetric MRI we do because we want to know whether the brain shrinks or not. In Alzheimer's, over 18 months, the brain shrinks substantially. So if we see no shrinkage, it means that our drug protects nerve cells from dying. I need to point out here that this screening process with the 3 steps takes a minimum or takes 8 weeks, sometimes a little bit more. And that's important when we'll go to recruiting because it takes a long time between when you have a patient that have the correct MMSE and then have that patient into going randomization because they have gone through the p-tau, through the plasma biomarkers, and through the volumetric MRI. So this is the study we proposed to the FDA and we are conducting right now. It is a 6/18-month study, which means that we actually have an analysis of the data at 6 months and then one at 18 months. As I just mentioned, we are looking for MMSE, we're looking for p-tau217, and the primary endpoints is ADAS-Cog13 and ADCS-IADL, plus volumetric MRI and biomarkers. We are allocating 380 patients to placebo and 300 to buntanetap, a total of 760 patients. And this is a more schematic diagram of the study. We started the 6-month arm. By next summer, we should have treated 760 patients for 6 months. And by fall, we will have the readout of the 6 months data. The patients remain blinded. They continue the study for an additional 12 months. And in summer of 27, we should have treated 760 patients for 18 months and then open the blind. And again, when the data actually confirms or negates that the drug is disease modifying, file an NDA. So the FDA cleared us for this study, and we are actually conducting it. This is our target. The target is 100 sites. We think we're going to open about 90, sorry. We want to screen 1,500 patients and randomize 760 to fulfill the patients we need. Recruitment is about a year, and the study is 18 months. The dose is 30 mg and placebo. And this is where we are today. So we have selected 84 sites, 52 are open. We have screened to date 220 patients. We have had 88 screen failures, which is a screen failure rate of 38%. Now, the data I need to explain is that we have enrolled 760 patients, but only 11 are on drug. And that is because of the 8-week delay. The 760 just started or are somewhere in the 8-week piece, and the 11 are actually on drug. Nobody has discontinued, and nobody, of course, has completed. I have shown this before. It's a nice diagram that shows the difference between disease symptomatic, disease modifying, and what we know about our drug and what we don't know. So the green line is the natural disease progression. Patients get worse over 18 months by about 5.5 points. That's an average. And if you treat them with Aricept, which is a symptomatic drug, they will get better for 3 to 6 months by about 1.5 points. But then they get below baseline, they get worse, and eventually they look just like patients that were never treated. They go back to natural disease progression. The blue line is Leqembi and Kisunla. These two drugs do not improve cognition. What they do, they slow the progression. In fact, they slow the progression by 30%. So the blue line actually has a slope that is slower and flatter than the green line. So the further you go in treating these patients, the more the distance between the natural disease progression and the disease modification. On top, the purple line, that is us. The solid line is what we know. At 1 and 3 months, we are over 3.5 points better than baseline, which is 2.5 times better than Aricept, the standard of care. After that, the line gets open because we really don't know how it's going to progress. It could stay as we have depicted here. That's our hope, because you may notice that it has the same slope as Kisunla and Leqembi, which means it is disease modifying, but at the much higher level because it improves cognition to get started by 3.5 points. Now, of course, it doesn't have to do that. It could come down. If we are really lucky, it could stay even higher, but I mean, don't hope for too much. But there is a reality that it could come down and just be symptomatic. Now, even if it was just symptomatic, I want to point out that short term, it would increase cognition 2.5 times better than the standard of care, which is nothing to sneeze at. And now let's go to Parkinson's disease. As I was telling you, in Parkinson's, the best result we have was really not in the UPDRS, which is the Parkinson scale. It was in cognition. And so in order to minimize our risk of doing an incorrect study, we decided to be conservative and do a study in Parkinson's patients that have cognitive decline. And they are two groups. They are grouped under Lewy body dementia, but there is dementia with Lewy bodies and Parkinson's disease dementia. The patients we have in our study and we have analyzed are actually most likely Parkinson's disease dementia patients. So we proposed 2 studies to the FDA, one for DLB and one for PDD. And again, we are using 30 mg. And we showed them the design. We were expecting a response by last week, and the response we got was we're overworked and understaffed, and we'll get to it. We don't know when. So we don't have a response. Now, realistically, I don't expect them to be terribly negative because there are no drugs for Lewy body dementia. There are no guidelines for Lewy body dementia. And we probably have the absolute best data in Parkinson's disease dementia cognition. So I don't see any problem for them to say yes. Now, we also, a lot of you want to know, when do we do the open label extension study? And yes, we again have asked the FDA. We have actually not asked. We have submitted a protocol, and we expect them to approve the protocol. And that is expected by later this month, so maybe in a few days, because there isn't that much left in a month. And let me show you what we sent to the FDA and what we hope we're going to get. So here you see Parkinson's disease dementia and dementia with Lewy bodies. The lowest hanging fruit for us is Parkinson's disease dementia. With 300 patients, we can do a pivotal study and actually get statistically significant data if we use the same patients we found in our Parkinson's disease study. And so that's, once we have the money, the most likely for us to actually conduct. We also asked for the open label extension. And here there is a little something different. We will, of course, ask, or we will invite all the patients that have already been in one of our Parkinson's studies. But we also decided to include patients with DBS. And the reason is that patients with DBS are routinely left out of clinical studies because nobody knows how they respond to drugs since they have electrical stimulation in the brain. But since this is an open label study, we can take the risk that they don't respond. And so this study will have between 500 and 700 patients. Half of them, or as many as want to come from the previous Parkinson's studies, and then the other half, or as many as we can get, that have DBS. And that is it. Thank you very much. I'm open for questions.
Thank you, Dr. Maccecchini, for that comprehensive update on the company's progress. Like you mentioned, we will now move to the Q&A portion of the webcast. And I already see some hands up there. Thank you so much. First of all, we are going to address some questions that have come in written, and then we will address the questions live that I see people raising their hands for. So let's get started with a few questions that we have written. Question 1, Dr. Maccecchini, if the AD data are positive, when do you expect the drug to be on the market, and how long does it take for the FDA to approve it?
So it's a difficult question because I was telling you that we will open the blind for very few people next summer, analyze the data, have something in fall, and if it's good, file an NDA. But the FDA is changing. On one hand, they're saying that they want to make approval faster, especially for drugs that are safe, especially for drugs that have a huge unmet need. So there is a possibility that we get fast track. There is also a possibility that overworked and understaffed is still a reality. And then I really don't know. But if everything goes as planned, it would be about a year after we have the data. So fall of 27 for symptomatic and fall of 28 for disease modifying.
Thank you, Maria. All right, next question. Will there be an open label extension after the 18-month completion of AD study?
Yes, absolutely. After the Alzheimer's study, we will attach the open label, yes.
Perfect. All right, we'll move to the next question. I see more coming in as we speak. Is it possible to begin the regulatory submission process while the planned 18-month confirmatory trial is still ongoing?
I hope so. As I just said, it's not 100% clear where the FDA is going, but yes, Maccecchini says so. Yes, so let's hope he is right. Maccecchini is the new director of the FDA. Commissioner, I
Okay,
think is the name.
perfect. Thank you for that. All right, moving on to the next question that we have here. Do you have a set date for Parkinson's clinical trials, including open label?
We are desperately, and I really mean with all my strength and every minute I can dedicate looking for money. Okay. So right now, I haven't really totally given up on open label this year, but we need to raise money, and it has been very difficult.
Okay, thank you. When do you anticipate full enrollment for AD trial, and how long after that will it take to obtain meaningful results?
So we hope to be done by the end of the year. You remember I said we have so many patients screened, but only so few in the clinic. In 8 weeks, 60% of those will be actually on drug. So the beginning has been painful because A, not all your sites are open, and B, it takes 8 weeks to recruit somebody. And then you have 40% failure on top of that. So we really hope to be done by year end and then treat for 6 months.
Perfect. Okay, we're going to answer one more question before going to live mics. What is the funding status for all these anticipated programs?
Well, I just said I'm trying very, very hard. Yes.
Good. Okay. I saw several hands. We're going to open the floor for live questions now. Thank you, Maria. I see Jason McCarthy. You can feel free to open your microphone. All right, you're live, Jason.
Hey Maria, thanks for taking the questions. A question about the MRI approach that you had mentioned, and I think I caught it right. You're looking at MRI and imaging of the brain to show symptomatic modifications at 6 months, or is this for disease modification over 18 months? And if so, do you need to correlate that to specific biomarkers, and can it fluctuate with water content and kind of all of the above? Can you just elaborate a little bit more on that?
Well, I think it's all of the above. And we just had a meeting with KOLs. And the trouble is that PET has been approved. p-tau217 has been approved, but as a biomarker, not to show disease modification. So PET has been approved as a biomarker and for disease modification. p-tau217 has only been approved as a biomarker. So when we finish and show the difference between p-tau217 here and p-tau217 there, and brain-derived tau here and brain-derived tau there, and neurofilament light here and neurofilament light there, if we see a difference, which we believe we do because we saw it in our other studies, and can then also combine them with volumetric MRI and of different parts of the brain, because it's not just the ventricle, it's a whole bunch of different parts. I'm not an expert at that. Well, we hope that's good enough. And the FDA hasn't told us it's not good enough. But in terms of PET, there is a precedent. In terms of what you're doing, there is not.
As far as the ongoing phase 3 Alzheimer's program, do you expect the 38% screening failure rate to continue at that rate? Kind of what are some of the key elements that are driving those fails? Is it very specific and defined inclusion criteria and exclusion criteria? And is it comparable to other large-scale trials, or you just can't compare to their different drugs, different inclusion criteria, et cetera?
Well, our calculation and our expectation was 50%. Okay. The 38% corresponds very, very nicely with the p-tau ratios that we saw in our phase 2/3. Now, interestingly enough, we actually don't have that, well, we don't have that many p-tau and MMSE, a few MMSEs, but we also have a lot of kidney and liver and kidney failure people. So we exclude them for something that's unrelated, but I'm told you don't want to have kidney failure in your study, so.
Got it. And this
But
is
to
more,
compare with others, Leqembi and Kisunla, and also prasinumab or gantenerumab, they had 80% screen fail.
got it. And this is more of a broad current, I used to call it current events question. Have the changes at FDA and leadership in certain and departures in certain departments at FDA have any potential impact on Alzheimer's or Parkinson's drug development that you can anticipate? I know the change that occurred just last week with Prasad coming
Yes.
in. He was very against the aducanumab approval in 2021, I think it was, and issues with some other drugs that were controversially approved.
So the interesting thing is that I was down at an FDA meeting and I actually talked to Macri and I talked to Jackie. And what I postulated is that Alzheimer's disease is being evaluated the same, whether it's a biologic or whether it is a drug. I don't think it's fair that if you have a biologic, you have to jump through less hoops, especially since biologics have more side effects than if you have an oral drug. And they both agreed. They said they're considering doing a neurodegenerative group that specifically looks at diseases that take a long time, take long studies, and where we are starting to look predominantly at disease modification. Now, Jackie left. I have no clue what's going to happen. I don't know.
Got it. Great. Thanks for taking the questions.
Thank you so much, Jason. All right, next we'll hear from Michael Gopes. You are live. Mike, you can just turn on your microphone. Michael, there you go.
Thank you. Maria, good to see you. Question on the phase 3 trial you've entered on Alzheimer's, and just looking backwards on the last trial you did, you were much better on the symptomatic improvement than the current drug Aricept. And you said you'll see if there's going to be disease modification as you go further out 18 months.
Mm-hmm.
But I wondered if you could just clarify the current drugs that show that disease modification over a short period of time have a totally different mechanism and treat the disease after it's already affected the brain, is my understanding. And your drug treats it solves some of the issues in the proteins in the brain. So if you could just clarify between your drug and the drugs on the market, and if it's better, whether you expect to see a better result.
Yes, I definitely expect to see a better result. And the short studies we did show a better result. And the reason is that in the brain of an Alzheimer and or a Parkinson's patient or an ALS person or a Huntington's person, there is more than one neurotoxic aggregating proteins. Pharma, like Leqembi and Kisunla, they are targeting A beta, which is plaque. They're not targeting anything else. But if you look at the brain of these people, they have plaque, they have tangles, they have alpha-synuclein, they have Lewy bodies, and they have TDP-43. And if you look, they probably may have some prions. There were some studies about that, but have not been reproduced. So if you just remove one neurotoxic aggregating protein, you still have the others. Just think if you have 4 killers, you kill one, the other 3 still kill you. And that is the beauty of our drug. It inhibits them all.
Okay, that was my question. Are you seeing biomarkers on all of the proteins in the brain that it shows improvement with buntanetap? In
animals, yes. In humans, not all of them have been discovered in the brain, in the blood, sorry. In brain of animals, we can reduce every single one of the proteins I showed you. And in CSF in humans, we have seen a reduction in a lot of these proteins. In plasma, it's hard to do. We only have A beta 42, we only have brain-derived tau, we have some taus, some p-taus. But there is not much else.
But you're showing improvement in more of these than any other drug on the market or at this point.
I don't know, because I cannot really compare apples and oranges. See, Kisunla and Leqembi have shown that they reduce plaque. I don't see plaque. I measured P217, and P217 is supposedly correlated with plaque. When I have the 6 and the 18-month study, I will know if it goes down and it correlates with plaque. Right now, it's an assumption.
Okay, thank you.
Thank you very much, Michael. All right, we'll move to Dawn Tice. You're welcome to open up your microphone, Dawn.
Okay, can you hear me?
Yes, you're
Yes.
live.
Okay, my question is for someone who has not participated in or completed any of your previous Annovis trials, when do you expect to have another clinical trial available for brand new participants?
You put me in a tough spot. You know, I would like to do all the studies that are needed to get this drug approved for any indication it may be working on. I don't know. As I just presented to you, you're going to go for the low-hanging fruit. Number one, we have no money for Parkinson's studies. Okay, let's be very clear about that. And I'm not going to do a study for which I have no money. So what I presented, though, is that the first study we will do in Parkinson's is the low-hanging fruit, which is cognition. So if you don't belong to the Parkinson's disease dementia patients, you're not going to be in that one. Now, if the stock goes up, if we can raise an additional 70 million, I'll do that study.
Okay, thank you very much.
Thank you, Dawn. Okay, Antoine Romanos, we'd like to hear from you. You're welcome to turn on your microphone. Antoine Romanos. Okay, you're live.
Hi, Maria. Thank you for the call. My question is about the pivotal phase 3 study that in the chart I saw that so far you've only enrolled 11 patients out of the 760, and you're projecting to be able to have full enrollment by the end of the year, which is, 6.5 months from now. How do you see us achieving that?
Well, that's why I was trying to explain that there are 760 patients screened. And somehow they're stuck between the 8 weeks. Actually, I'm not sure that number is correct, but there are 280 patients screened that are stuck between those 8 weeks, which means that in the next 8 weeks, we will have over 200 more patients. And then
Why are they stuck?
Because it takes 8 weeks between when we say hi, and we have done all the tests to know that they're early Alzheimer's disease.
Got it. So basically enrollment's on track and you don't foresee any issue with getting to 760 patients by the end of the year.
Well, we are a little slower than I hoped, okay? But we have been picking up quite nicely in the last month. So yes, I think we'll be there.
Okay, got it. Thank you.
Thank you very much, Antoine. All right, we'll hear from Christy Castro. You're welcome to turn on your microphone, Christy. All
Hi,
right,
thank you for taking my question. So I've heard from a couple of actual participants and with Parkinson's disease in the past, and they have said they had such great improvement. And those of us who have Parkinson's disease have been waiting for, the new recruits, those of us who are new to get in, like the previous person asked about. So it sounds like it's going to be quite a ways down the road. If being hopeful, I'm wondering in the future, what would be the soonest time frame that you think could be possible for it to be approved for all of us with Parkinson's disease, you know, to go through our doctors and receive this drug? Are we talking 5 years, 10 years? If everything works out? I
No,
know you don't
I
have a
hope
crystal
not,
ball.
okay? I hope not, but then I have been kind of optimistic a lot. So let's say, or let's assume that the 6 months Alzheimer data is good. And I really believe it is because we are replicating the previous study to a T. And if the previous study, that group worked as well as I showed you, and the forest plot proves that it wasn't cherry-picked, then the present study at 6 months should work. Well, the stock will go up, we can raise money. You know, people say, why can't you raise money if your company is worth 50 million? Because I can't. I can raise 20% of who we are. I cannot raise 200% of who we are. So it's just not possible. So yes, let's say that symptomatic works, we can raise 100 million, and then we can do the Parkinson study. Since we are not doing the studies, we are talking to the FDA ongoing to get things in line for when we can do the study. Shouldn't take that long to do it. Then maybe 6 months later.
Okay, thank you.
Wanted to know what's on the market. On the market, it would be
yeah.
3 years after we start the Parkinson study.
Okay. All right. So if we include that Parkinson's study, could take a year or two, then we're talking probably 5 years or so. Thank you for giving us a realistic timeline on that.
Yes.
Thank you, Christy. Appreciate it. All right, we'll hear from Greg Gallo. You're welcome to turn on your microphone, Greg. You're
Hi,
live
Maria.
now.
Hi, Maria.
Hi.
What I was wondering is when I think I heard you first talk, it was 10 million in funding you were requiring. Now it's 70 million.
No. Different studies. Okay. Open label, 10 million a year. I was saying an 18-month full-fledged Parkinson study that shows symptomatic and disease modification. Different
Okay,
studies.
so 70 million for that. Now, let me ask you one other thing, Maria. Can we lobby our state and local government officials to provide funding? They're taking all this funding away from all these institutions. Couldn't some of that be rerouted to you? And have you also went to investment firms to see whether there's any money available there?
Yes. Yes to all of the above. Yes.
Okay. Well, I know you're trying. So we'll have to hope that you're successful.
Or open black market in Africa, something like that.
Yeah. Okay. Thank you.
Thank you, Greg. All right, we'll hear from Dennis Beckman. Dennis, you're welcome to turn on your microphone. You're live now.
Thanks very much. Well, first of all, I'm really pleased to hear you including some DBS people in your study. What's this? How do you recruit people for your open label DBS cohort?
We talk to PIs and universities that do DBS. And Cheng, I think she is on the call. She actually has really good contacts, and we have started to talk to them. And they're very happy to give it their patients because DBS patients just are always left out.
Indeed, that's true. And who is that person you were talking about?
Cheng Feng, she works for Annovis.
Okay.
She has the
Thank you.
contacts with the PIs with the DBS sites.
Thank you.
Thank you very much, Dennis. All right, Brendan Duffy, you're welcome to turn on your microphone.
Oh, great. Hello, Maria.
Hi.
I'm I'm I have Parkinson's. I was in your study, and I was not taking the medication, and it was surprisingly, I felt amazingly better because the placebo effect is so powerful. My question to you is, there was some time ago I saw that study you did on the axons, how the speed of the axons was greatly increased. Is there any additional information you can share about that? Because I found that to be particularly interesting.
Well, we didn't really do any more axonal transport. However, what we have tried to do in humans, where you can't really look at how fast a nerve cell fires, we have looked at biomarkers in CSF that show how well a nerve cell lives and works and produces. And in humans, we see an improvement in nerve cell health. Now, realistically, I don't know exactly how that helps us. But in animals, we know it is about axonal transport.
Okay, thank you.
Thank you
Good
very
luck.
much, Brent.
Thank you.
I see Greg raised his hand again. You're welcome to open up your mic, Greg. Greg Gallo.
I just had one other thought. I know a lot of us, maybe a lot of people don't know how they became Parkinson's or Alzheimer's susceptible. I know in my case, and that's why I say the government again, that they ought to be lobbied. I used a solvent and I talked to a doctor I know in Australia, actually stopped to see us last year.
Yeah.
And the stuff I used at work was, he called it nasty stuff.
Yeah.
It claws your gut. And then I believe when you get to be at my age, or 60, 65-ish, your blood-brain barrier can't fight off the bad stuff anymore. And that's when, not everybody's that way, but that's when Parkinson's starts, and I imagine Alzheimer's in some people. That's why I was saying before that the government ought to be funding this, because they failed us.
Well, that is blaming the environment, blaming chemicals, blaming shots to the head. A lot of things make us susceptible to neurodegeneration. And you are 100% correct. Chemicals contribute more to Parkinson's than to Alzheimer's. Hits to the head contribute more to Alzheimer's than to Parkinson's. But then certain hits, just like football players or boxers, they contribute to chronic traumatic encephalopathy. And then the reality is, did you eat too many carrots? Were you protein insufficient? You eat too many carrots, you don't get protein. It seems like, and then there are all the reasons about infections with viruses, with bacteria. They're all true. Every single one of them is true. There are so many things that can injure the brain. And you are correct that whoever did it should pay for it. But they're going to get away with it not. And you know why? Because there's so many things that cause Alzheimer's and Parkinson's. There's so many things that cause MS. Plus the simple fact that you breathe air, it causes something that it's really hard to prove. And you see the lawsuits that are going on about Roundup, they're going to go on forever.
Yeah. A bunch of us used to use a solvent for a rubber cleaner when things were electromechanical, called Fedron. And it was nasty stuff. As I said, I talked to this doctor that's a neuro researcher, also happens to be a pathologist, a toxicologist is what I was looking for. So, okay, well, I think I know the path I want to take here.
Which is
Contact local officials and government officials and start lobbying them to funnel some of this money they're saving to worthwhile causes.
Thank you. I really would appreciate that. Because sometimes I think that more money goes to pharma. Lilly gets grants of $60 million to $70 million for its studies, and I don't.
Right.
Doesn't Lilly have more money than I do?
It's not fair.
Yeah, but if I say that, we're never going to get anywhere. It's just called keep going.
Yeah. Okay, Maria, thank you.
Thank you, Greg. Okay, we have time for maybe one or two more questions. Thank you so much for your hands, everyone. Let's hear from Robert Braun. You're welcome to turn on your microphone, Robert.
Hi, Maria. Appreciate this Q&A session. My main question is, for us, longtime investors, that's really what I've been since 2021. We want to know what we can do to help in this. We know there's a financing issue. Is there anything that you want the general public to know of something that we can do to potentially help?
Investing obviously is the only way to get the stock up so we can raise more money. I really believe that the drug works. I showed you a lot of studies, and then there is not one study we did where we had the opposite effect. And you can look at everybody else. When their studies don't work, they don't work.
Mm-hmm.
So, we are just putting one foot in front of the other. The market will get better. Raising money will get better. We'll have better data. Somebody will stop shorting us someday, sometime, and we'll be okay.
Okay, thank you. Appreciate it.
Thank you, Robert. Dick Sundrum, you're welcome to open your microphone. You're
Hello,
live.
Maria.
Yep.
Can you hear me?
Yes, I do.
I just basically want to thank you for everything you've done. I think the question I was going to ask the last few people have kind of covered it. I was just kind of wondering what patients and the public in general could do to help. And I guess the answer is just hope we can get our congressmen, our government to funnel more money to you guys, so that I think what you're doing is wonderful. And I just wanted to basically thank you for what you're doing. And when I've sent you emails asking you questions, you respond so promptly. And I just think it's great. And just want to thank you.
I honestly care about patients. I am in this because I really want to protect nerve cells from dying. If nerve cells don't die, you don't have the symptoms. If nerve cells stay alive, if they keep firing correctly, we can actually get rid of all neurodegenerative diseases. And that's what I'm in it for. Look, I'm getting old, okay? And I'm going to hang in until it's done.
Okay, we appreciate that. Thank you again.
Thank you, Dick. Okay, we'll take one more question from Justin Higgins. You're welcome to turn on your microphone, Justin.
Can you hear me okay?
Yes, you're live.
Fantastic. Thank you. So I want to follow up a little bit on what Robert was saying. I joined you on your last call, and I posed a question to you in that call. I'm hoping that we can make some progress on this call. The question I presented to you last time was, there's plenty of people on these calls, myself being one of them. I've actually made quite a bit of money on your stock in the last 3 months because I've been buying it at an unrealistically low level, and it's done very well for me. It's gone from $1.10 to, I think it was 3 and change this morning. And there's a lot of other people like that. Now, maybe their overall cost bases are underwater or not, but they are continuing to build their positions. As we build these positions, they can become uncomfortable at sometimes when we don't see progress, especially from the pharmaceutical world. So my first question is, the conversations you've been having, are you having them with investment oriented companies, or are you having any direct conversations with pharmaceutical companies? And if it is the latter, would you be willing to discuss who those companies are offline, not on this call, obviously, because it's a public call?
So we did both. I mean, right now I'm talking to banks. Last year, when we had the data, most of the data I showed you, we actually, Mark White, who is a board member, but was doing marketing at Pfizer, he helped me, and he contacted 41 companies, all the big ones. They all took a phone call. They pretty much all said, we don't know if it's disease modifying or symptomatic. We don't care about another symptomatic drug. It's 2.5 times better. We don't know if it's 2.5 times better. It could just be a fluke that it's that good. So come back when you have more data.
Okay, so that basically is what I expected you to say. And the only real way you're going to make progress with those larger pharmaceutical companies is if we're able to set up meetings where we can convince the management of those companies that your data is better than it's been presented. Would you say that you've had some trouble presenting your data in the past?
It's not better than we presented it. We have studies that show very clearly that we have 3.5 points improvement, and Aricept has 1.5 to 2. I didn't invent that. That's the way it is. But it's one study, one large study and 2 tiny studies. So what if we have better, the data is better than reality, and when we reproduce it, we're worse than Aricept. But if you want, but I'm happy to talk to you about how to present it better, because we were pretty much rejected, and basically we were told, show us more data.
That's fair enough. And there's lots of ways to skin a cat, as they say. So I think progress can be made there. My last question is, and I wanted to make sure that I had this correct, because I asked this question the last time, and some investors that I work with also said, we're not sure if that is what she meant. In the last previous study, there were some endpoint misses, okay? And we understand why those misses are there. You explained that very well. It took a while to get it all out, but it's been out there now. Your stock took a pummeling because of those misses, and that's pretty obvious. Now, my understanding of this 6-month endpoint that you are presenting is you are doing what large pharmaceutical companies do when they have endpoint failures in their studies. You are teeing up what you believe has already proven to work in the prior study. Is that correct?
100% correct, yes.
So the odds of the 6-month study going very well would be more likely than not likely because of the endpoint development and the design of the study itself.
Yes, I would give it 99%. Okay, give it
That's great.
100%.
Thank you, Maria, for that. I really appreciate it. I'll be buying lots and lots of your stock. Can't wait to buy more.
Thank you.
Thank you, Justin. And thank you, everyone, for your insightful questions. Sorry that we're not able to take more at this time, but you're always able to contact later. Thank you all for participating in today's webcast. So we'll close our Q&A section for now. We truly appreciate your engagement and your interest in Annovis. As a reminder, a replay of today's webcast will be available on our website shortly. Thank you once again for joining us. And thank you, Maria. Have a great day, everyone.
Thank you very much, and goodbye.